The next one that we are going to talk about is the continuous cultivation. And the continuous cultivation is when you have a fermenter and then you put a fresh substrate in, but at the same time, you take the fermenter content out as well. See that? There's flow in there and it's flow out there. So that's why continuous cultivation is an open system. And yes, the media is continuously added, okay? But at the same time, you withdraw the fermenter content out as well. Okay. The point in continuous concentration is that the flow rate that is going equal to the flow rate that is come out. This one, they are equal. So with the equal flow rate in and flow rate out, you have also no change in volumes. Okay, it's no change in volume, but it is an open system since you have change in mass. Okay, mass going in your system and coming out the system. And the mass balance on the cell is often used to derive what happened in a continuous culture. Okay, let's see at this mass balance equation. The changes of something in this case, I give an example of the change in biomass. Okay, the change in biomass is such that in the flow in, you feed the media in. Your media contain only substrate. There's no biomass in it. Okay, and there's no product in it. I mean, theoretically, for the continuous cultivation, okay, you only have substrate in. So that means that your biomass is not in the flow in. That's why the flow of biomass in is 0 here. Okay. But once the substrate goes in the fermenter, there are cells in fermenter. So there's a cell growing in the fermenter. That means that when you draw the content of the fermenter out, you have the cell out of the fermenter as well. So that is why you have the Fx out with the negative, because you're drawing something out from the fermenter. You're drawing biomass out of the fermenter, to be exact. Another term is that the X is being grown in the fermenter here. So that's why you have the biomass generated as a positive term here. Okay, and since we assume that there is no cell death, okay, because all the cell is being used for something, so the consumption of the biomass is 0. So basically, you are left with 3 terms: this term, and that term. Okay, for the mass balance of the continuous cultivation. Now, one thing about continuous cultivation is if you keep putting the substrate in and you're drawing the fermenter content out at the same rate. At the beginning, the changes of things in the fermenter, you will notice the change of things in the fermenter. But after a while, okay, the content in fermenter start to be stable. Okay. Start to be stable, and when things start to be stable, that means that if you take samples, you don't see the change in biomass, you don't see the change in substrate concentration, and you don't see the change in product formation. What we call that state is a steady state. So since there is no change over time, but there's still growth, there's still cell growth, there's still product being produced, substrate being used. But if you take sample at different times, you don't see the change because it's being drawn out this way. Okay. In this case, at the steady state, the change of a thing in the fermenter, let's say, is equal to 0. Okay, there's no apparent change. So that means that this term become 0. Then you left with this term and that term, okay? And you equate the equation, you rearrange the equation, and this equation is what you get, okay? And from our previous lecture, you know that dx dt, the change in biomass which is being produced, equal to mu x. And the flow out here, okay, is equal to this term times x. Okay, we will talk about this term, which is the dilution rate, in the next slide. Okay, and for continuous cultivation, the dilution rate equal to a specific growth rate. This is the important point for the continuous cultivation. Okay, and this D is the dilution rate. Okay. So let's see the unit a little bit on these two terms. Okay, the unit of mu we know is per hour or per time. Okay, let's say time is hour, and x is generally gram per liters, right? So that's for mu x, and this x is also gram per liter. So the unit of D is the same unit as the mu, which is per time. So let's see what dilution rate is. Okay, by definition, dilution rate is the volumetric flow rate that goes of the stream that goes into the fermenter, which equals to the flow that is out of the fermenter, and the V, okay, the A, and the V is the fermentation volumes. It's not a fermenter volumes, okay? It's a fermentation volume, so how much media you use, that is the fermentation volumes. Okay, the term dilution rate is given because there is a flow into a fermenter. So apparently the content is dilute. Okay, you have more flow, it's more dilute. See, you have more flow, more dilute. And if you look at the picture, you see that you see the flow of the general continuous culture. You have something flow in, something comes out, which are equal. The thing that goes in normally contain a substrate. Okay, the substrate get consumed in the fermenter while cells is generated and product is generated. So that means that in the fermenter you have substrate, product, and cell. So these three things goes into the flow out of the fermenter as well. That's why you have these three things out as your product stream. So this is the harvesting line as well for the continuous culture. So seeing the flow like this, you should conclude that the concentration in the outstream is just the same concentration as in the fermenter stream. Okay, so like I said earlier, in continuous culture, apparently after a certain period of time, a steady state will be reached. So when we have a continuous culture, we start the fermentation as a batch, okay, just to get the cells to grow, okay, and then you feed in and take samples periodically until you see no change in the concentration of the three things that, as we said, and that is when you know that it reaches a steady state. Okay, this is how a typical plot of continuous culture look like. Okay, so if you see the plot that you have like a substrate concentration, biomass, and product concentration versus a dilution rate, you should immediately assume, okay, actually you can say that it is the profile for the continuous culture. Okay, so in this case, okay, of the continuous culture, if you increase the dilution rate this way, so you increase the dilution rate, and let I write here that the dilution rate is A on V. Okay, if you have the same V, increasing the dilution rate means that you increase the flow rate in and out of the fermenter as well. And another thing that I should write here is that in continuous culture, mu equal to D. So seeing this graph, what you see here is the increasing in dilution rate. That means that at each point when the steady state is reached, a cell growth rate increase to the right-hand side of the graph here. Okay. What happened here in continuous culture is that when you have a small dilution rate, your cell grows slow. Your cells grow slow. It has more time to use the substrate. Okay, because the flow is small. Okay, the flow is small. Small dilution rate means the flow is small. So the cell have more time to use substrate. So at the in the fermenter and in the harvest line, you see that the substrate concentration is low because it has been used. Okay. When you increase the flow, that means that the substrate has less time to stay in the fermenter. Of course, you still have cells growing, but it have less time to use the substrate. So when the steady state is reached, you get increasing in the substrate concentration and smaller biomass concentration. Okay, and the typical curve for the continuous culture profile is that as you increase the dilution rate, the flow rate is more. Of course, you get the cell with a higher growth rate, but the concentration of the cell itself would decrease as well because and the substrate concentration increase because the substrate has less time staying in the fermenter. The cell has less time to use it. It reached steady state, but it can't use like all the substrate. So to the point, okay, to the point where the flow in is so fast, the cell cannot keep up, even though it grows fast as well, okay, because mu increase as well. Even though it grows fast as well, but it can't keep up with the time, the small time that the substrate goes into the cell. Eventually, it will reach a point called the washout. Okay. The name says washout. It means that everything is washed from the system. When everything is washed out of the system, that means that there's no biomass in there. The substrate that you put in stays there. it hasn't been used because it goes so fast. It goes into so fast, go out so fast, the cell doesn't have time to use it. The cell doesn't grow, and it's being washed out. Okay. So if you look at this curve, you see that immediately that, okay, if the substrate line at the washout is 12, you know that you put in the substrate of 12 grams per liter, and at washout, what you put in comes out. That's it. Okay. So there's no cell in the fermenter because it's been washed out. And this would give you a one important kinetics parameters. Okay. At this washout point, for this example, is washout at D equal to 0.3 per hour. And that thing is also your mu max. Okay. This is an important point for continuous cultivation as well. So determine the mu max. Look back in our previous lecture. You can do the semi-log plot and find the mu max from the batch cultivation. But mu max is closer to whatever you are interested in, more in a continuous culture, because this is the physical things that actually happen. So the dilution rate that at the washout is the same as the mu max. Okay. This is a theory graph. Okay. This is in theory that follows the monod kinetic. Okay, and if you remember, is monod kinetic is dx dt equal to mu max s over ks plus s times x. Okay, and that thing is the mu. Okay. So this is a typical curve for the monod kinetics. In the actual continuous culture, which this is from a research paper. This is from a research paper. It's actually my PhD paper, and you see that it doesn't really follow the monod kinetics because it has the product inhibition and substrate inhibition things happen as well. And this is how it actually looks. This is for lactic acid bacteria fermentation. Okay, you see that we feed in lactose at about 40 grams per liters, and we do the fermentation, changing the dilution rate. Okay, and the washout, if you extended this, it goes to about 1. So the maximum specific growth rate for this specific work is about 1 per hour. Okay. And this is a good estimation if you want to do like a kinetic modeling. Okay, that equations that you see, this could be an like initial values for you before like you do the mathematical calculations and then get the actual mu max from the equation. Okay, a point to note here is that washout, the point that washout occur is when the dilution rate is higher than the maximum specific growth rate. So we can estimate the maximum specific growth rate at the which is the maximum dilution rate as well. Okay, so that is the main point for continuous culture. You should understand what dilution rate is. You should understand what happened in the continuous culture. You should understand that in continuous culture, the important point is a a steady state. Okay. Now the last variation of the cultivation mode is the fed batch cultivation. Okay. In fed batch cultivation, as the name said, is the batch cultivation that has been fed. Okay, so that's why it's called fed batch. So basically with fed batch cultivation, you start off the fermentation as a batch fermentation, okay? And then you feed something in. So that means that a fed batch cultivation is an open system, okay? It's an open system because you have the flow in, so it's an open system. Okay. And in fed batch, as in the batch, you harvested at the end of cultivation. Okay, it's not like continuous culture where you have the flow out of the fermenter that you harvested it all the time. Okay, so it's harvested once at the end of the cultivation. So with this method of fed batch cultivation, the volume may change during the cultivation because you have the addition of substrate. Okay. Actually, hypothetically, fed batch cultivation, it's not supposed to have a volume change, but it's like in theory, in the ideal situation. But we'll talk about how we feed in and how the volume could change or how the volume might not change. Okay, but first have a look at the change, the mass balance in the fed batch cultivation. Okay, so in fed batch cultivation, you can do the balance of, you can see the mass balance also for the substrate, the biomass, and also the product. So for the substrate, the change of substrate, okay, which is here, depends on two terms, okay? We have 4 terms for the mass balance. Since there's something coming in to the system, so you have the flow of substrate in, okay? You don't have the flow out of the system, so that's why your flow out is 0. And you're not supposed to have a substrate generated in a fermenter, so your change of substrate from the generation is 0, but the substrate is being used in the fermentation, so the substrate consumption, the change in substrate for the consumption stays there with the negative size. Okay. Similar concept for the biomass and product. Okay, I only give example of the biomass here. Okay, so the change in biomass depends on only one term. So what happened in the fermenter only. Because why? Because in this feed line, you don't normally have biomass feeding in. You often feeding in substrate or feeding in the media to supply more into the fermenter. Okay, so the biomass is only generated here, and similar things happen to the product. Okay, for the fed batch cultivation, okay, there are 4 strategies to feed the thing into the fermenter. Okay, these are the 4 strategies that we use to feed things in the fermenter. I want you to have a look at the 4th one first, okay? This 4th one is what the theory of fed batch should be. Okay. You start off the fermentation using batch fermentation, and you feed in only the substrate. Like if you are using glucose or you're using sucrose, you feed in the glucose or sucrose solution with a high concentration. High means very high. Big substrate is very high. So start the fermentation as a batch. You add the substrate that have a much higher concentration than that in the starting media. For example, you might start your fermentation with only something like 10 grams per liters of sugar, and then you add a substrate, probably like 300 grams per liter of sugar, something like that. So that at the end, you are feeding in the amount of sugar that you want to feed into the system, okay? With these strategies, the volume shouldn't change that much. So we say the volume not change, okay? I mean, if possible, they probably want to just add the powder in and then mixing them. That would be the ideal case, but generally it's not happen. So you add in only a small amount. So you might start the batch fermentation at 5 liters, but you might add a substrate, only something like 50 ml or something like that, just to, so the volume change is not very significant. So you can say, okay, the volume doesn't change. Okay, so this is the ideal case for fed batch culture. However, doing this thing is pretty, you have to have a very good control of the flow, okay? So there are some other variations that has been used in the fed batch cultivation as well, and we are looking at those three now. So the first one that I want to introduce is that you add the media and the substrate, okay? So you start a fermentation as a batch, and then you start adding the same media. So you might start with like 20 grams per liters sugar and nitrogen sauce, and then you add the same thing into the fermenter. Using this method of feeding strategies, the volume will change. Definitely the volume will change, and it will change a lot as well, okay? So with this strategy, you might start a fermentation with like 20 or 30 or 50% of the fermenter volumes, and then you add a lot of amount just to make sure that you have enough substrate. You have the desired amount of the substrate that you want to feed into the system. Okay, and next one, okay. Next one is similar to the previous one, okay. You start the fermentation also as a batch, and you add only the substrate. For example, only sugar solution, which has the same concentration as in the starting media. So in the first case, you might add, wow, it will end again in 10 minutes. Okay. Okay, if this thing happens again, I don't know why, because I have already booked the staff up. If this thing happen again in 10 minutes, I'll ask you to do the same thing and using the same method, okay? I'll have a careful look next time we are doing things. Okay, in the first method, okay, you have your, for example, say sugar, and then you have your nitrogen here, and you add the same thing here. With this method, you start the fermentation having sugar and your nitrogen sauce and other thing for your starting media. But the substrate that you put here is only like a sugar solution, which has the same concentration as the sugar you start off with. Okay, so say if you have 20 grams per liter of sugar, this sugar solution that you add in is also 20 grams per liter, for example. And the third way that we do is adding the substrate that have similar to number 2 here, but adding the substrate that have higher concentration than that in the starting media, but not very high or as high as at this scenario. So for example, this one you might start off with the 20 grams per liter of sugar and your media, okay? And you probably add something like 100 grams per liter, which is only 5 times more concentrated, of the sugar solution into the fermenter. So these 4 feeding strategies are a little bit different, but it's in the same principle that you start off things with the batch fermentation. The volume depends on how you design your experiment. And you only add things in, you don't take anything out. And you only harvest it at the end of the fermentation. So that's why it is called a fed batch fermentation.